Hello, my name is Ekaterina Filatov. I'm a research assistant for Dr. Gray at UMBC. Within the field of energy metabolism, we are trying to understand how to mitigate obesity, a state where energy input outweighs energy output. The neuroendocrine factor PACAP is known to mediate thermogenesis. Brown fat is a thermogenic organ stimulated by the sympathetic nervous system. The outflow tracts of the sympathetic nervous system are believed to begin within the hypothalamus, where PACAP is also highly expressed, particularly in the ventromedial and paraventricular hypothalamus. Although experiments have shown that PACAP affects brown fat thermogenesis with impacts on the sympathetic nervous system, the mechanism of how PACAP acts on the tracts is unknown. We hypothesize that PACAP is expressed within those tracts located in the hypothalamus. To test our hypothesis, we use the following experimental approach depicted in Figure 1. PACAP EGFP transgenic mice with PRV tagged with beta-gal, a gift from Dr. Card and Dr. Enquist, were used to label the sympathetic nervous system. The virus was injected into interscapular brown fat depots of transgenic mice. Because the virus is retrograde, it will travel from the peripheral system back up to the central. Using immunohistochemistry, this design allows us to visualize EGFP and beta-gal expressing cells, representing PACAP and virus-infected sympathetic outflow tracts respectively. Serial sections spanning the ventromedial hypothalamus were cut. Post-immunohistochem images were stitched to show the hypothalamus as a whole. Figure 2a shows the area of focus and is an example of stitched pictures composed of micrographs. Looking at Figure 2b, we can see red fluorescent cells, allowing us to say that our surgical injections were successful. Using the tiled images, we were able to assess the structures listed off in Table 1. We present the preliminary analysis for one brain. This included rating the structures in terms of density of green EGFP and red beta-galactosidase expressing cells separately per structure. Now looking at the last column in Table 1, we are finally showing data that pertains most to our hypothesis. In this column, we show what structures had at least one cell where two of the fluorescent labels co-occur. Those results allow us to infer that PACAP is expressed within the outflow tracts of the sympathetic nervous system innervating the brown fat depot. Figure 3 shows a cell that has a co-occurring fluorescence. Most excitingly, we are able to see co-occurrence in the ventromedial, paraventricular, and lateral hypothalamus, although rarely. We plan to continue this study with a greater sample size, looking at more areas of the brain, and adapt this experiment to other adipose tissue depots. We hope to further elucidate PACAP's role in thermogenesis and to then have this knowledge applied in ways to mitigate obesity.